Welcome to the unit on eukaryotic microbes. In this unit, we're going to look at the different types of eukaryotic microorganisms that are found in different environments. While the majority of this course, we're really going to focus on bacteria, it's still important for you to understand that not all microscopic organisms are prokaryotes. And so we'll look at some of the different types of eukaryotic microbes and how they differ from one another. In this video, we're just going to go through a general overview of what a eukaryotic cell is as a bit of a refresher. Okay, let's get started. Remember, eukaryotic cells are typically the largest of the microorganisms, ranging from 10 to 500 micrometers in diameter. And unlike bacteria, which could be classified into a handful of distinct shapes, eukaryotic microbes come in a wide variety of shapes and sizes depending on the classification and species of the organism. Their defining feature is that they contain membrane-bound organelles, including a nucleus that houses the DNA. Organelles are beneficial to cells because they allow the cell to function more efficiently by compartmentalizing specific processes and allowing transport and movement around the cell. One of the key groupings of organelles is the endomembrane system, which consists of the nuclear envelope that becomes the endoplasmic reticulum, both the rough and the smooth, the Golgi apparatus, lysosomes, and the vesicles that travel between these organelles. The endomembrane system is important because it is used for secretion of proteins from the cell and for transporting materials into the cell through endocytosis and phagocytosis. Proteins that will be secreted from the cell are first translated on ribosomes of the rough ER, represented by these circles. Those proteins get translated and put into the ER lumen, such as this protein here. That protein will be packaged up in a transport vesicle and then get sent to the Golgi, where it travels through the various membrane stacks until it is eventually transported out into a secretory vesicle on the other end. Secretory vesicles then travel to the cell membrane, where they fuse with the lipid layer and release the protein either out into the environment, or this is also what occurs when we need to have proteins embedded in the cell membrane itself. The system also works in reverse, with endocytosed or phagocytosed molecules entering into a vesicle through the cell membrane, which will then transport them to the lysosome for digestion. Everyone should remember that mitochondria are the energy-generating organelle of eukaryotic cells, while chloroplasts are used for photosynthesis. While you've probably learned about chloroplasts in relation to plants, they are also found in algae, one of the eukaryotic microorganisms. The interesting part of both mitochondria and chloroplasts is that they likely formed by endosymbiosis, with an early eukaryotic cell engulfing a prokaryotic cell, which then lived inside it. The prokaryote helped generate energy and was provided a home inside the eukaryotic cell, and after hundreds of thousands of years, that engulfed prokaryote has lost the ability to live on its own and are now the mitochondria we observe in almost all eukaryotic cells. The chloroplasts form through a second endosymbiotic event after mitochondria formation, when a cell engulfed a photosynthetic prokaryotic cyanobacteria. The evidence for this endosymbiont theory can be seen in the two structures. Both mitochondria and chloroplasts have two membranes, an outer membrane and an inner membrane, seen here and there. These two membranes were formed because the outer membrane would belong to the original eukaryotic cell, while the inner membrane was the original plasma membrane that surrounded the engulfed prokaryote. Both these structures also contain their own DNA, both mitochondrial DNA and nucleoid DNA, that is circular in shape, just like prokaryotes. They contain their own ribosomes to do translation, and so the DNA is transcribed and translated into proteins that are unique and specific to the mitochondria and the chloroplasts. Additionally, both organelles are able to replicate themselves through binary fission. All of these are properties that are left over from their original prokaryotic state. We see further evidence in the chloroplast as they also contain the thylakoid inclusion bodies that are found in cyanobacteria today. There are specialized structures in eukaryotic cells, just like there were in prokaryotic cells. These are not found in all eukaryotes, but only in certain species. Eukaryotic flagella and cilia are similar structures, with both used for movement of the organisms. Cilia are shorter and more numerous than flagella. Remember that prokaryotic flagella rotated like a boat propeller, while eukaryotic flagella will move side to side in a whip-like motion. This occurs because of the inner structure of microtubules that are found within both flagella and cilia, that we can see illustrated here. These microtubules will grow long on one side, forcing the structure to bend sideways. They will then contract, and the ones will grow on the other side, forcing the structure to move in the other direction, forming that whip-like motion. It's the microtubules that are able to grow and contract that allow this whip-like motion to occur. A key to keep in mind as we talk about the eukaryotic microbes is that most are able to replicate both asexually and sexually. This is in contrast to prokaryotes, which only divide asexually, with one cell dividing into two. Eukaryotes can grow asexually through processes such as mitosis, where remember we would have one parent. That one parent would have to do DNA replication, so we get 2 copies of the genome. It then divides itself into 2 separate daughter cells. That is considered asexual reproduction because we only had one parent or one cell to start with. That's different than sexual reproduction, where instead you have 2 gametes that are haploid, so an egg and a sperm, that come together in fertilization to make a zygote, which is now a diploid cell. This alternating between haploid and diploid states occurs through something like meiosis, which is forming sexual reproduction. Sexual reproduction is important because it allows shuffling of the genetics of a population, increasing the amount of genetic variation. The remaining videos in this unit are going to walk through the different classifications of eukaryotic microbes, including the protists, fungi, and helminths. Of those, the most complicated classification is going to be the protists, because this is a historic classification that refers to a diverse group of microscopic organisms that just didn't fit into any of the other taxonomic groups. The organisms thought of as protists do not have a shared evolutionary history, as you can see from the many branches on this evolutionary tree. It's in contrast to taxa such as animals or fungi or plants, which only have one branch on the tree showing their shared evolution.